The two scientists Nernberg and Mattei wanted to know what specific codons coded for what amino acid. They went about this by creating a synthetic mRNA strand made out of a uracil subunit. They called it the poly-U mRNA strand. They put a poly-U mRNA strand in a test tube along with 20 amino acids, one of which would be radioactively labeled. The test tube was also filled with stuff required for translation. They allowed the reaction to continue, and then they filtered out the protein that would be created from the rest of the reactants. They then tested the protein for radioactivity, because if the protein was radioactive, that means that it used the specific radioactive amino acid. If it was not, then they tried a different amino acid and radio-labeled that one. They tried this with poly-U, poly-C, poly-A, and poly-G. And they got the results that phenylalanine was coded for poly-U, proline was coded for poly-C, and lysine was coded for poly-A. Poly-G was attempted but did not work. Nernberg then went to another scientist named Lieder in order to figure out what other codons and amino acid matches could be made. They knew that codons came in groups of 3s and were coded for in groups of 3s, so that there are 64 unique codons. For this argument, let's say that they were trying the UGC codon. They would put all the machinery required for translation, and they would also put radioactively labeled tRNAs. Let's say this top one is cysteine, cytosine, and the top one was tyrosine. The bottom one was tyrosine. They would allow the reactions to continue, and then they would filter over nitrocellulose paper. Nitrocellulose paper is important because it binds ribosomes. The ribosomes would also contain the mRNA sequence and the complementary tRNA sequence with the amino acid. They then would check the nitrocellulose paper for radioactivity. For the cysteine one, they would find that the nitrocellulose paper would be radioactive, given that UGC codes for cysteine, and the tyrosine one would not, because it does not code for it. They tried this with all 64 and found that 61 out of 64 codons could be determined. That is because 3 of the codons are stop codons and do not code for anything.